Bean bunny evolution. Remember, you're not necessarily using beans. I use paper balls. You can use whatever you have available. Modeling gene frequency change or evolution in a population by natural selection. In this activity, you will examine natural selection in a small population of wild rabbits. Evolution on a genetic level is a change in frequency of alleles in a population over a period of time, meaning frequency of alleles is how often you come across those alleles. So from generation to generation, how often changes. Breeders of rabbits have long been familiar with a variety of genetic traits that affect survivability of rabbits in the wild, as well as in breeding populations. One such trait is the furless rabbits, or naked bunnies. This trait was first discovered in England by W.E. Castle in 1933. The furless rabbit is rarely found in the wild because the cold English winters are a definite selective force against it. That means it's too cold for rabbits to survive without fur. In this lab, the dominant allele for normal fur is represented by a capital F, and the recessive allele for no fur is represented by a lowercase f. Bunnies that inherit two big F alleles or one big F and one little f allele have fur because the big F is dominant, while bunnies that inherit two little f's have no fur. You need to have two of them that have no fur because that's recessive. The purpose in this activity, you will model evolution by natural selection of the gene frequencies of two alleles in a population of organisms. You will see, to reword this, how the gene frequency changes from generation to generation. You will also calculate the gene frequency of the alleles for each generation and then graph the frequency of the two alleles over 10 generations. I made the graph for you. You just have to plug in the numbers. Materials. Each lab group will need the following: a bag, so brown paper bag, small. It doesn't have to be that. I used a hat in my video, whatever you got. 50 beans, red, 50 beans, white. Again, use whatever you got. I used blue and pink pieces of paper. A marker and 3 petri dishes. I used styrofoam cups instead of petri dishes, and a marker to draw on them. Procedure: working with a partner, which you are not doing now because I made it easier so you don't need a partner. I did a lot of work for you. Read the following problem question for today's lab. How does natural selection affect the gene frequency over several generations? From your previous knowledge about natural selection, evolution, and genetics, answer the problem question on your student answer page in the space marked hypothesis. That is going to be on a separate page on Google Classroom. This is actually last year's Google Classroom. Pay no attention to the dates. On the Google Doc, in your hypothesis here, how does natural selection affect gene frequency over several generations? So what do you think will happen to the frequency of the alleles? State your hypothesis in an if-then format. If this happens, then this will happen. Include your prediction regarding how you think natural selection will affect the gene frequency in a population. State what you would predict about the frequency of big F alleles and little f alleles in a population of rabbits after 10 generations, where little f bunnies, homozygous recessive bunnies, are selected against and do not survive. The red beans, or I use blue paper, whatever you're using, represent the big F allele for fur, and the white beans, I used pink paper, so whatever you're using is what you're using, represent the little f allele for no fur. The paper bag, and I use a hat, represents the English countryside where the rabbits randomly mate. Using the marking pen, label one petri dish big F, big F. I did this on a styrofoam cup for homozygous dominant. Label a second one big F, little f for heterozygous, and label a third one little f for the rabbits with homozygous recessive genotype, the furless bunnies. Place 50 dominant alleles and 50 recessive alleles in the paper bag or whatever you're using and shake it up. That is the rabbit's mating. Please note that the frequencies have been chosen arbitrarily for this activity. So yeah, it's not always going to be 50-50 to start with. That's just how we're starting. Without looking at the beans, select 2 at a time and place the rabbit or bean pair into the appropriate dish. Big F, big F, big F, little f, or little f, little f. For example, if you draw out one recessive and one dominant, place both in the big F, little f dish. Continue drawing up pairs until all the beans have been placed in the appropriate dish. To determine the number of individual rabbits produced for each phenotype, count the total number of alleles in the appropriate dish and divide this number by 2 since the individuals represent a pair of beans. Meaning, if you pull out 2 dominant alleles, you pull out 2 alleles, that counts as 1 rabbit. If you pull out 4, that counts as 2 because every rabbit has 2 alleles, right? If you have 8 alleles, that counts as 4 rabbits. Record your results on the data table. The little f bunnies are born furless. The cold weather kills them before they reach reproductive age, so they cannot pass on their genes. Place those alleles from little f aside before beginning the next round. I dumped them in the trash can. You will not count those in the number of little f alleles column. If they're dead, they can't reproduce. You cannot count their genes. Count the surviving big Fs and the surviving little f alleles that were placed in each of the furred rabbit dishes in the first round and record the number in the data table under the columns labeled numbers of big F alleles and numbers of little f alleles. Total the number of big F and little f alleles for the first generation. I already did that for you. And record this number in the column labeled total number of alleles. I already did that for you. You just have to find out how many big Fs and how many little f's you have. Place the alleles of the surviving rabbits, big F and big F little f, back into the container and mate them again to get the next generation. Repeat steps 6 through 9 to obtain the generations 2 through 10. So everything you just did, repeat it for all 10 generations. Make sure to take turns so that each partner has a chance to select the beans and record results. You don't have a partner, doing it all yourself. That's why I did your math. Determine the gene frequency of big F and little f for each generation. I already did that for you. If there is time, your teacher may have you record your group's frequencies on the board. We will not do that. Complete the conclusion questions on the student answer page. So underneath your hypothesis, you come back to this page and answer these questions. There are 8 of them. Graph your frequencies. I set that up for you. If your teacher requested, plot the class totals. We will not be doing that. Everything else is already set up for you digitally. So I remade this graph for you, and we made that for you, and I remade all this for you. It is all right here and right here. All right, so if you have any questions, comments, or concerns, please feel free to contact me via email or Google Classroom are fine. All right? All right. Good luck, ladies and gentlemen.